In the early 1900s, sheep farmers across rural Europe dealt with a sickness that made no sense. A healthy animal slowly changed. It lost weight, moved awkwardly, and rubbed its wool raw against posts and fences, as if an itch lived under the skin. Then it worsened, week after week. Nothing brought it back. Every case ended the same way. The farmers called it scrapie, and the name fit the most visible sign, not the cause. What frightened people most was the timing. Scrapie did not tear through a flock overnight like a fever. It waited. Animals seemed fine for months, sometimes longer, and then the decline began. By the time a farmer recognized the pattern, it was too late to separate the sick from the well. Some flocks lost only a few. Others kept seeing new cases appear long after the first deaths, as if the disease hid in plain sight. Farmers tried the usual explanations. They blamed bad feed, harsh winters, parasites, even breeding lines that looked cursed. But none of it matched what happened on the ground. Scrapie showed up on farms that changed nothing, and it avoided others that shared the same grazing land. It also seemed to travel with animals, yet not in a simple, predictable way. One fact stayed solid. Once the wasting started, the outcome never changed. Researchers stopped treating scrapie like a curse of the pasture and started treating it like a testable problem. If it spread, something had to move from one animal to another. So they took material from sick sheep and introduced it into healthy ones by inoculation. The result came slowly, but it came. After months, then longer, the same wasting, the same loss of control appeared. It meant the disease could be passed on purpose. That success brought a new kind of certainty and a new kind of frustration. Certainty because a transmissible agent had to exist. Frustration because every ordinary suspect kept disappearing. Under the microscope, there was no consistent bacterium, no parasite, nothing that looked alive in the usual way. Researchers tried to grow it, stain it, and isolate it, the way they did with familiar infections. The experiments kept giving the same answer. Something infectious was there, and it stayed hidden. Inoculation also changed what contagious meant. Scrapie did not need a visible invader to act like an infection. It behaved as if it carried instructions, even when no organism could be found. That forced scientists into an uncomfortable middle ground. The disease clearly passed between animals, yet it refused to fit the categories of known germs. The only way forward was to probe the agent itself, not by assumption, but by brutal tests of what could destroy it. By the middle of the century, the obvious suspect was a virus. So researchers treated scrapie material the way they treated viral samples. They filtered it, trying to trap anything with size and structure. The surprise was immediate. Even after passing through filters meant to hold back viruses, the infectious effect remained. Whatever carried scrapie did not behave like a normal microbe. It acted smaller, tougher, and harder to pin down. Then came the tests that should have ended it. Radiation was a standard way to cripple life by shredding DNA or RNA, the genetic instructions every known virus depended on. Scientists hit the scrapie agent with doses that ruined ordinary viruses. Yet the agent still caused disease. More treatments followed. Chemicals that damaged nucleic acids, heat levels that weakened typical infections. Again and again, infectivity persisted. The results did not fit the rulebook. Each experiment narrowed the options, but none produced an answer. If the agent carried genes, those genes seemed strangely protected or strangely absent. If it was a virus, it ignored the very attacks that defined what a virus was. Researchers faced a growing contradiction. A transmissible disease that resisted the tools designed to expose and destroy genetic pathogens. The scrapie agent looked infectious. but it did not look alive in the usual way. In the 1950s, attention shifted far from European sheep pens to the highlands of Papua New Guinea. There, a strange illness appeared in reports from field doctors and patrol officers. It looked nothing like malaria or influenza. People began to stumble, their hands shook, they lost control of movement, then speech, then life. The local name for it, kuru, spread with the case numbers. It clustered in specific communities, especially the Fore people. As clinicians observed kuru more closely, patterns emerged that felt unsettlingly familiar. The disease moved slowly, over months, and ended the same way every time. Autopsies showed brains damaged in a distinctive, sponge-like way, holes where healthy tissue should have been. That kind of degeneration echoed what scrapie had done in sheep. Yet this was humans. There was no obvious germ, no fever, no pus, no sign of a conventional infection. The question tightened. What kind of agent did this? The turning point came when researchers tested whether kuru could be transmitted the way scrapie had been. They took brain material from victims and inoculated it into primates, then waited. Years passed. Then the animals developed the same creeping loss of coordination, the same fatal decline, and the same spongy brain damage. It meant kuru acted like a transmissible neurodegeneration, not a toxin or a curse. A human disease had joined the scrapie puzzle by behavior, by pathology, and by proof. By the time kuru was tied to an infectious process, doctors elsewhere faced their own baffling cases. Creutzfeldt Jakob disease, or CJD, looked like a lightning strike of dementia. People lost coordination, speech and control, and then died. Under the microscope, the brain did not show a clean infection or a tumour. It showed holes, a sponge-like wreck. The pattern felt familiar now, and that recognition mattered. Researchers started lining up these rare illnesses side by side, scrapie in sheep, kuru in humans. CJD appearing mostly at random, and sometimes in families. Other strange syndromes joined the list, like Gerstmann-Streusler-Scheinker disease and fatal familial insomnia. The names sounded unrelated, but the damage matched. Rapid decline, tiny vacuoles in brain tissue, and an outcome that never changed. Different faces, same ending, and possibly the same cause. That shift turned scattered medical oddities into a single category: transmissible spongiform encephalopathies. The key idea was simple and unsettling. These conditions did not just resemble each other. Some of them spread by exposure to diseased tissue, even across species. The old boxes of neurology and infectious disease no longer held. A family had been identified, and once it had a name, scientists could track patterns, compare experiments, and finally hunt for one underlying agent. By the late 1970s, the central problem still refused to go away. Whatever caused these brain-wasting diseases kept acting infectious, yet it did not behave like anything biology recognized. Into that confusion stepped Stanley Prusiner, a young neurologist who focused on a simple question. If the agent resisted treatments that destroyed genetic material, then what survived? He began breaking infected brain tissue down, fraction by fraction, chasing the part that still carried disease. Prusiner pushed the material through harsh purification steps and compared what remained with what still infected animals. Over and over, the strongest infectivity stayed tied to a protein-rich fraction. That result sounded almost impossible at the time because proteins were supposed to be tools, not blueprints. DNA and RNA carried instructions. Viruses used those instructions to copy themselves. Yet here, infectivity kept following something that looked like protein, even as the usual signs of nucleic acid faded. To name what he thought he had found, Prusiner coined a new term, prion, short for proteinaceous infectious particle. The claim went further than a label. It proposed that the infectious agent needed no DNA, no RNA, only a protein, somehow able to reproduce its effect inside a host. It was a direct challenge to the most basic rule of infection, and it turned a decades-long puzzle into an even bigger one. How could a protein behave like a germ? The prion claim hit like an accusation against the rulebook. Many scientists resisted it because it seemed to erase the one thing every germ needed: genetic instructions. The debate turned fierce, and it turned personal because careers and whole fields rested on the old logic. But the argument did not stay philosophical for long. It moved into test tubes, where the agent faced one practical question: what exactly carried infectivity? Again and again, researchers attacked the infectious material with tools designed to destroy DNA and RNA. They used enzymes that chopped nucleic acids apart and treatments that should have wiped out any virus-like genome. Infectivity kept surviving. Then they did the opposite. They targeted proteins. When the protein component was damaged or removed, infectivity collapsed. The trail kept pointing to one place. The infectious punch followed a particular host protein, especially when it was misfolded. That pattern forced a new kind of explanation. If no detectable DNA or RNA rode along, then replication could not mean copying a genetic code in the usual way. Instead, the evidence fit a stranger chain reaction. A malformed version of a normal protein pushed other copies of that same protein to fold the wrong way, too. In this view, the host supplied the raw material, and the misfolded shape supplied the instructions. It was a rethink, not a tweak. By the mid-1980s, a new brain disease appeared in British cattle. Animals that once seemed healthy started to act strangely, then lost coordination, then died. Veterinarians called it bovine spongiform encephalopathy, or BSE. Tests showed the same kind of sponge-like damage seen in scrapie and related human disorders. This time, though, the setting was modern farming, and the numbers climbed fast. Investigators traced the outbreak to a routine practice meant to save money and boost protein. Remains from slaughtered animals got rendered into meat and bone meal and fed back to cattle. If even a small amount of prion-contaminated material entered that system, it did not get sterilized away. It got recycled. Each turn through the feed chain amplified the agent and widened the spread across herds. Once the route became clear, the implications landed hard. A disease that took years to show itself could still move quickly through an industry built on shared feed and nationwide distribution. Officials imposed bans and began destroying affected cattle, but confidence had already cracked. People questioned what else might have entered the food supply. BSE turned prions from a lab argument into a public alarm with real economic consequences. In the 1990s, a new fear followed the cattle crisis into hospitals and homes. Young, otherwise healthy people began developing a strange brain disease that looked like CJD but did not behave like the classic form. It carried different symptoms and a different pattern of damage. Doctors called it variant CJD. The implication landed hard. The animal outbreak had crossed into humans. Once that link seemed plausible, the question stopped being scientific and became practical. How many people had been exposed, and where did risks still hide? Governments tightened food rules, removed high-risk tissues from the supply, and tracked animal feed more aggressively. But prions forced a wider rethink. Infection did not need coughing or contact. It could travel through products, procedures, and systems built on trust. Blood became a new worry because prions did not behave like familiar germs, and silent incubation lasted for years. Blood services restricted donors tied to exposure and changed processing to cut risk. Surgery posed its own problem. Standard sterilization did not reliably eliminate prions, so hospitals revised instrument handling, quarantined sets, and adopted harsher decontamination. Public health had faced outbreaks before. This one required rewriting the rules of prevention. By the time the alarm over cattle and human cases settled into policy, the central idea no longer sounded impossible. The infectious agent did not need genes, it needed shape. A normal body protein could fold the wrong way, and that wrong fold could push other copies into the same dangerous form. Replication meant conversion, not copying a code. The old rules of infection had stretched, and biology had to stretch with them. With that shift, the mystery became something scientists could test in detail. Different prion strains could be explained as different misfolded shapes, each producing its own pattern of damage and timing. Species barriers stopped looking like magic and started looking like a tight molecular fit, sometimes breached, sometimes not. Laboratories tracked how prions moved through tissue, how long they persisted, and why heat and chemicals failed when they would have worked on ordinary germs. In the present day, prions sat in a category of their own, protein-based, self-propagating misfolding, watched closely even when case numbers stayed low. Surveillance continued in livestock and in hospitals, and research pressed for faster detection, safer decontamination, and ways to block conversion before brains were damaged. The world ended up with a clear mechanism, but the lessons stayed unsettling. What began as baffling scrapie in sheep had rewritten what infection could be.